In this session, we are going to look at biology, and the topic we'll dive into is translation. But before we can get to look at this topic, let's try to understand the grammatical interpretation of the word translation. To translate is just to change maybe the language into another language without altering the meaning. That is the meaning of translation. When you have a person, let's say, speaking in Spanish, and the people that he or she is communicating to cannot hear Spanish, however they can hear English, then has to be a translator. There has to be someone who is going to be interpreting that Spanish into English so that the message can be communicated well to the audience. So this is basically what we are looking at. Now, in biological terms, that is the grammatical meaning of translation. Now let's look at the biological interpretation of translation. What is translation in biology? So we've looked at the central dogma of molecular biology. Central dogma of molecular biology. This is the background where we are coming from. We said for proteins to be manufactured, there has to be DNA. Now this DNA cannot be converted directly into RNA. It has to make at least some copies of itself. So DNA has to be made into DNA again. It has to make a copy of DNA. Now the process by which this DNA makes a copy of itself, this is called replication. It just replicate is to make a copy. Then this DNA has to make a copy of RNA. It has to be transcribed, and the process is now called transcription. So it is just a certain portion, a certain segment of DNA that needs to be transcribed. So to be specifically, it is transcribed to mRNA, messenger RNA, and this process is called transcription. Now this mRNA is the one now which is going to form proteins. Then proteins has to be made. Now the process by which proteins are made from mRNA, that is what we are calling translation. And in this session, this is our focus: translation. So let's define what translation basically is. So we say translation, this is just a process of making proteins. So it is just the process of making proteins from mRNA. So that is translation. So from messenger RNA. So we've defined what translation is. Now let's look at some of the important things. There are 3 things which are needed for this translation to take place. For the proteins, in short, to be synthesized, there are 3 things which are needed. So protein synthesis okay, protein synthesis needs three things. Number one, it needs the ribosomes. The second thing it needs is RNA. Then it also needs amino acid. So these are the three important things which are needed for protein synthesis: ribosomes, RNA, and amino acid. So now let's look at these ribosomes. When you're looking at ribosomes, you're just talking about the organelles which are involved in protein synthesis. Organelles involved in protein synthesis. Okay? Now, we need to understand that these ribosomes, they are non-membrane bound organelles. No wonder you'll find that the ribosomes are found in both eukaryotic cells and also prokaryotic cells because they are non-membrane bound. Prokaryotic cells do not contain membrane bound organelles. So that is very important for us to understand. And the other thing I want us to understand is that this ribosomes is made up of two subunits. So it has two subunits, and these subunits are made up of RNA, rRNA, which is ribosomal ribonucleic acid, and also the proteins. Now let's look at the two subunits of ribosomes. Subunits of ribosomes. So we have two types of cells: the eukaryotic cells and also the prokaryotic cell, or prokaryotes and eukaryotes. So let's have small subunits there. So maybe I can rub here. Let's have small subunit, large, then let's have combined here. Okay, so we are saying we have the small subunit, the large subunit, and the combined. So in eukaryotes, the small subunit has 40S. The large one has 6S. But this is not the addition. This is not mathematics like to find the combined then you add the two. No. You just add the 20 on each. So 20 plus 60S is 8S. So the large one is 8S. To find the subunits in prokaryotes, you simply subtract. So small, for eukaryotes we had 40. So 40 minus 10, you subtract 10 on each and every subunit. So we'll have 30S. For the large, 60 minus 10, we'll have 50S. Combined, 80S minus 10, we'll have 70S. So it is very important for us to understand that. And this S we are talking about is the Sedberg unit. Okay? It is just the rate at which the ribosomes sediment. It is just the sedimentation rate. The rate at which the ribosomes are able to settle on the bottom of the test tube. Okay? when you carry out centrifugation. That is what we call the Sedberg unit. So it's very much important for us to understand that. Now let's look at this RNA. We'll discover that we have three kinds of RNA. We have what is called messenger, which is abbreviated as mRNA, messenger RNA. We have what is called the transfer, abbreviated as tRNA. We have what is called the ribosomal, abbreviated as rRNA. So this ribosomal RNA, this one is abbreviated as rRNA. Let's look at their function. When you're talking about the messenger RNA, this carries the genetic code. Okay? This one just carries the genetic code or the genetic information. When you're talking about the code, it is just a set of instructions which determines the type of the protein to be formed. So that is what is called the genetic code. So, and the genetic code has what is known as the codons. It is made up of codons. Those codons are just a group of three nitrogenous bases. That is what is called a codon. We can have, for example, the start codon AUG, which is made up of the adenine, uracil, and also guanine. So that is what we need to understand about the messenger RNA. Then when you're talking about the transfer RNA, tRNA, this transmit what is known as, or transport what is known as the amino acid. So it transports amino acid for peptide synthesis. This is what we need to understand about transfer RNA. Then when you're talking about the ribosomal RNA, this is the one now which is part of the ribosomes that make proteins. So this one combines with ribosomes, the ribosomes in order to form what proteins. It is just a part of ribosomes which form proteins. Okay, so now let's try to understand that translation takes place in 3 steps. Okay, so let's look at steps of translation. So basically it takes place in 3 steps. That is, the first step is initiation. The second step is elongation. The third step is termination. But there's also another stage, or another step rather, which takes place other than this. It is called post-translational modification. When proteins are made, they have to be modified. Okay, let's say this is the 4th step, which we are calling post-translational modification. Let's look at insertion. Let's start with insertion. Insertion. So when you're looking about insertion, we understand to say that translation takes place in the cytosol or in the cytoplasm, right? So what insertion is, we can have a strand of RNA, so 3' 5 prime to 3 prime there in that way. So to begin insertion process, this mRNA has to locate what is known as the small subunit. It locates the small subunit in the cytoplasm. So let's say the small subunit is there. Okay, so that is the small subunit. So this mRNA will have to locate this small subunit. Now, once this small subunit is located, there's something which is going to help this mRNA to locate or to bind rather to this small subunit. And that thing which helps this mRNA to bind to the small subunit, that is what we call the insertion factor. So with the help of the insertion factor, this mRNA will bind to the small subunit. So with the help of this insertion factor. Now, we need to understand that the small subunit moves in this direction, in the same direction, which is 5 prime to 3 prime. And this small subunit, as it tends to move, it locates now the start codon. So we are saying, as it tends to move, it locates the start codon. The start codon is AUG. So AUG is the start codon. We are talking about the codon. A codon is just a group of 3 nitrogenous bases. So AUG meaning adenine, guanine, and U, it is uracil. So adenine, uracil, and guanine. So we need to understand that when this start codon is reached now, when the start codon is reached, what is going to happen? It is going to send signal to tRNA. So once the start codon is reached, signal is going to be sent. So it is going to send signal now. It is going to send signal to tRNA, so that this tRNA can find the amino acid which corresponds to this codon. And in that case, because AUG is the start codon, when you use the genetic code, we will find that the AUG, the codon which corresponds to AUG, is simply methionine. So this tRNA will find, or bring rather, what is known as methionine. This is the amino acid it is going to bring. But this methionine is for eukaryotes. But when you're dealing with prokaryotes, prokaryotic cells, it is not methionine in the start codon. Okay, I mean the start amino acid, the amino acid which is blocked by the start codon. This is for eukaryotes. But when you're dealing with the prokaryotes, it brings what is known as methionine. So for prokaryotes, what is brought is formyl or F-methionine. So this is what is blocked by the start codon in prokaryotic cells. Now let's understand the structure of tRNA. Structure of tRNA. So tRNA has this kind of structure. So let's try to understand it. So there's something like this. Okay, so it has something which looks like this. So what we need to understand here is that this tRNA has some parts. This tRNA has some parts. So the longer part there is the 3' end. And we need to understand that the 3' end is where the amino acid are going to attach. So amino acid will attach at the 3' end, the longer end. Then we have this 5' end, the shorter one. Then we have the T arm there, the transverse arm, another T arm there. Then we have this arm which is inside. This is known as the transverse arm. Transverse arm. Then at the bottom here, this is the anticodon. So at the 3' end, there are some amino acids. You will find that, there are some codons. There's a codon which is ACC. This ACC. So what you're going to have is this. So at the 3', there is this codon ACC 5'. Okay? And we need to understand that the 3' there is the acceptor. So this is which is attached to the amino acid. So we have this ACC, which is a codon at the 3' end. So it's very important for us to understand this. Now let's look at what happens. So we have this strand of mRNA. Then we have the small subunits there. We have the small subunit. And AUG was found. So the tRNA has now come. So the tRNA has come. Like that. So this tRNA has brought the amino acid which corresponds to this. So it has brought the amino acid. So because there is AUG, the anticodon there on tRNA will have a certain complementary codons. So A pairs up with U, so that will be U. U pairs up with A, G pairs up with C. So this is the anticodon, which is complementary to AUG. So it is UAC. So in short, what I'm saying is that the codon, this codon, will pair up with the anticodon. Okay? So when this tRNA brings now the amino acid, this large subunit is going to attach. So what is going to happen is this. So the large subunit then, it is going to bind now. So this is what is going to happen. So this is the small subunit. The large subunit now has come to bind. The large subunit will bind now because the tRNA has brought now the amino acid, and this is the first amino acid which it brings. So we have the E site there, the P site, and the A site there. So this is 5' to 3'. So what is going to happen is that, so this is the large subunit, okay, then this is the small subunit. So this first amino acid which is brought, it is brought to the P side, okay. So the first amino acid which is brought to the P side. So what we have is this. So the first amino acid there will be brought to the P side, okay. So we have these sides, the E side, which means the exit side, okay. We have the P side, which is the peptide transferase, the A side, which is the amino cell, okay. So we need to understand that each of these sites have 3 nucleotides. So the amino acid which is brought there, the methionine, is brought, it is the only amino acid which is brought direct into the P side. The rest first comes to the A side, okay, methionine. So that is what we need to understand. So let's say the UCC is brought to the A side, right? UCC is brought to this side, the A side. Let's say UCC comes there. UCC is brought to the A side. So if we use the genetic code, we'll find that UCC corresponds to serine. So look at how to read the genetic code. When reading, you start with the first bases, the second and the third bases. So we'll look at how to read. So UCC, when it is brought, this is a codon. So the amino acid which it corresponds to, it is the serine. So serine is the one which is going to be brought into the A side, okay. And all you need to understand is that when this large subunit, when the tRNA has bound rather to the amino acid, when tRNA binds to the amino acid, it is formed like, it forms a complex. It forms a complex. So what is going to form is a complex. So tRNA, when it binds to the amino acid, a complex is going to be formed. That complex is the one which we call amino acyl tRNA complex. So it is very important for us to understand that. So it's very important for us to understand this complex which is formed. tRNA plus amino acid forms tRNA, amino acyl. So let me write this word properly. It is amino acid. Then tRNA complex. Okay, but this serine, when you're talking about serine can be brought by different codons. For example, the UCC. So we just consider the third position. At the third position, serine can be brought by different codons, which just differs the third position, at the third position rather. So for that reason, that third position is called the Ribo, because it can be brought by different codons, which just differ at just one position. And we need also to understand that the peptide transferase okay, let's look at this for better understanding. Let me draw this strand 5 prime 3 prime there. Then of course we have the small subunit. The large subunit has attached. We have the E site, the P site, the A site. So we have this site. And also that side. So we have that. Okay. So let's say this is what we have. So what is going to be happening is that this amino acid which has been brought to the A site, they use the amino acid which is serine. It is going to be connected now. It is going to be connected to the amino acid at the what? At the P site. Because we need to understand that it is only the P site which is able to grow. Okay? The growing chain is only in the P site, so it is going to be connected to this P site. Now, there is a certain enzyme which helps in forming a bond between this amino acid in the P site and the amino acid in the A site, and that is what we call the peptide transferase. The peptide transferase. So this peptide transferase is the one now which is going to form the peptide bond so that the growing chain can continue in the P site. Okay? And one thing I want us to understand is that it is only the ribosomes which are able to move. So it is only the ribosomes which are able to move, and they move in the 5' to 3' direction. And as they are moving, they move with the what? They move with the 3 nucleotides. Okay? They move with the 3 nucleotides. So it is very important for us to grasp that concept. So let's look at how it begins to grow. So we have 5', 3' end. We have the small subunit. We have the E site, the P site, the A site, the large subunits like that. So what is going to be happening is this. This is the A site. So when the amino acid has now bound to this, so we have the amino acid there, to the growing chain, you discover that the A site is empty, right? The A site is empty. So the tRNA then will be thrown out through the E site, which is the exit site. That tRNA will be thrown out. Okay? So the amino acid now begins to build. Those are the amino acids. They begin to build, and that is the large subunit. So another amino acid is going to be brought by a certain, let's say GUG, by a certain codon. It brings another amino acid. Okay, it brings another amino acid there. Again, this amino acid attaches by a certain enzyme peptidyl transferase. So the peptidyl transferase provides this peptide bond for this amino acid to attach to those which are in the P site. The chain begins to grow like that. So we'll discover that again it is empty. The tRNA is thrown out through the exit site. Again, it reads, the A site reads another amino acid. The amino acid connects to the P site, to the amino acid in the P site via the peptidyl transferase, just like that. So the chain begins to grow. So at the end of the day, what you're going to have is this. So 5' 3'. Then we have the small subunit, the large subunit. So at the end of the day, you discover that the process is just continuing until a certain point. So this is the P site that has begun to grow. It is growing, just like that. Okay. So this is growing now is what we call elongation. It is elongating. Okay. It is forming a large chain. So this is the chain. The chain continues. The chain continues to grow. But there will be a certain point. There will be a certain point when this A site is going to read a release factor. There will be a certain point when this is going to read what is known as a release factor. When the A site reads this release factor, so once the release factor is read, you need to understand that they are what are known as the stop codon. So there will be a stop codon, and they are basically UAA. We have UGA, the stop codon, and UAG. So we have the UAA, UGA, and UAG. So when these stop codons are read, okay, of course there will be a release factor which is going to come to the A site. And once this happens, once the release factor comes and binds at the A site, of course termination will stop. So it means that termination now has occurred once the release factor is read on the A site. So what are we saying? In the presence of release factor, binding at the A site, termination occurs. So we need to understand that once this is read, termination does what? Occurs. Okay, now let's look at something which I didn't talk about for a certain reason. We need to understand that before translation occurs, sorry for that. So before translation occurs, the tRNA has to undergo what is known as tRNA charging. Okay? So this tRNA RNA. This tRNA will undergo what is known as tRNA charging. Okay? So when you're talking about tRNA charging, it is just the addition of the amino acids to the 3' end of the tRNA. So when you have this tRNA like that, when you have this tRNA, we have the 3' end there. So when with tRNA charging, it's just the addition. The amino acid are going to be added to the 3' end for this tRNA to undergo what is known as tRNA charging. So that is tRNA charging. So it happens before translation commences. tRNA has to be charged. Some amino acid has to be added to this tRNA in order for it to be what? Charged. Okay? And this happens by the assistance of a certain enzyme. The enzyme which helps in tRNA charging, it is called the amino acid synthetase. The enzyme which assists is amino acid synthetase. You see, with an enzyme has, let's say, tase at the end, it has tase, tase. It means that this enzyme uses energy. Okay? It uses energy, which is ATP. But if it doesn't use energy, it would have been amino acid synthase. But this enzyme uses energy. So it is very important that we take into consideration that point. And there's what is known as amino acid activation. When you're talking about amino acid activation, it is just the addition of energy to the amino acid. So there is what is called amino acid activation. So with amino acid activation, we are saying it's just adding the addition of ATP, which is energy, to the amino acid. That is what we call amino acid activation. Now let's look at the last process which we talked about, which is known as post-translational modification. So with modification, when you're thinking about modification is just the improving of something, right? To develop something, to add, to make some slight changes, to correct something, to make some correction. That is what is known as modification. So when you're dealing with modification, we're trying to add something, we're trying to correct something. So with post-translational modification, we may add some phosphate group by the process known as phosphorylation. So we have what is known as phosphorylation. So phosphorylation, we are adding just the phosphate group to the chain which has been formed now. So this one, we are adding the phosphate group. That is phosphorylation. We can also add some methyl group by the process known as methylation. We can methylate it. Okay? With methylation, we are adding the methyl group. that is methylation. We can maybe add some glucose to the chain which has been formed by the process known as glucosylation. Glucosylation, we are just trying to add glucose to the chain. That is glucosylation. Or maybe we can try to add some enzymes which can activate, such as we have an enzyme which can activate the inactive proteins that is known as the zymogens. So the zymogens, we can add some enzyme to the chain by the process known as protosylation or protosylysis. That is the common name, protosylysis. So this one, we are trying to add some enzymes like the zymogens. Or maybe we can try to add some lipids by the process known as lipidation. So with lipidation, all we are trying is to add lipids to the chain which has grown. Or we can add some nitrogen, let's say N as a tRNA, but that is even more complex. Okay, so this is what we need to understand about translation. Basically, that is what translation is. So in the next session, we'll talk about the genetic code. Then we'll be done with this topic. Thank you for watching.